Okay, in the next 10 minutes or so, I will going to share with you some of the tools that I really like and that they are widely used in this community, and potentially they might be helpful for you to explore data and use it for your own research. So once you have a high-C data library sequence, what do, right? A really popular tool that you can use in your desktop is called Juicebox. It's still from Lieberman's group. The idea is actually you can use this app. You just download Juicebox, and then once you generate your Hi-C library, you process it. You have your Hi-C file. You can load your Hi-C with Juicebox, and you can just double click and zoom in, zoom out. It's really fast, right? You can also see the inter and intro chromosomes interactions. They can also do the eigenvector value confirmation for you. I really like this tool. You can use it. You can potentially also add the one-dimensional tracks for you as well, so they can link different part of genome with the quantum looping event to chip-seq signals. So another really great resource is WashU Epigenome Browser. Again, it's invented by my close friend Ting Wang from WashU. So the advantage for WashU Epigenome Browser is actually beyond. So the Juicebox, that's a application. I think they began to also have a web application as well. But the WashU browser is really a browser based. It holds more than 10,000 genomic data sets from ENCODE and roadmap and lots of Hi-C data. The idea is actually you can see you can load your Hi-C matrix, you can manipulate the heat map, and then they also have this arc-like structure. Once you give the predicted looping events between the two loci, you can align them with these tens of thousands of tracks together. It's really a great resource for you to play with, right? And then another great web resource actually was invented by my group. This is called the 3D Genome Browser. This browser actually, we just checked, has been used for more than 1 million visits in the past few years from over 100 countries all over the world. So the idea is similar. We have preloaded, everything's preloaded. The annotations, I don't keep all the tracks of chip-seq raw signals, but we do have all their peak files. And then we preloaded more than 300 genomic Hi-C or captured Hi-C type of 3D genome data sets. And then all you need to do is just go to the website. It's 3dgenome.org. I hope it can appear on the screen. 3dgenome.org. You can check on your iPhone. You can use it on your computer. All you need to do is go to the website. It's specifically designed for Hi-C and type of 3D genome structure. You can just choose your cell type from the drop-down menu and type your gene, type your GWAS, type your genome loci. That's it, right? You just go there, choose your genomic regions, click submit, and you will have something like this. We can show you the Hi-C map. You can see this is a quantum interaction between this region to the other region. Right? So for this 3D genome browser, you can also show multiple tracks as well. So in this case, you can see, you can even load different tracks. In this case, we're showing the human and the mouse for the similar gene. You can see the conservation of the tag structures. And we can also host built-in session for both Epigenome browser and UCSC genome browser, so that it can, again, align your chip-seq tracks with the Hi-C data matrix. Again, so far we have loaded 30, 40 Hi-C paper data sets, more than 300 of them. Every data is processed using the same pipeline. Loops are called, use Pikachu, and tags are also called with our DI models. So you are more than welcome to come to our website and use this resource. Again, so one of the community I really have to stress is this NH40 nucleosome projects. It started about 5 years ago, and it just the phase 2 just started last December, right? So for this consortium, we have more than, I think, 30, 40 investigators from all over the world, and people are studying the 3D genome structures using both the genomics assay and a lot of imaging-based assay. And the idea is that people will compare different genome assay itself, and also compare the observations you can make from genomics with imaging. The idea is we'll see if we can have a great complete picture of the 3D genome. Of course, 4D here means really you also have another state, 3D plus time series, right? During differentiation or during health to disease transition. So that's where the 4D come from. So again, the 4D 40N has processed a lot of data. Many data are deposited to the data portal. They also have processed a lot of the Hi-C and RNA fish and the TSA-seq different data sets, right? You can go to the 40N portal. If you just search 40N portal, you can see the data. Again, all the data, you can download them. They are, DCC is funded by the 40N, so they have a lot of resource. All the raw files and the matrix and then the BAM files can be downloaded. So this is a very nice visualization as well. So again, you can go to 40N to download a lot of the pre-processed file as well. It's a great resource, right? All right, to summarize today's lecture, I know I give you a lot of terminologies and a lot of the technologies and concepts. This is something I think might be really important for you for your future research. 3D genome is really tightly linked with the gene regulation, right? And currently there are two different main approaches: is imaging-based versus a genomics-based. And even for the genomics-based, there's a ligation-based technologies, different C technologies versus non-ligation-based technologies. And of course, compartment A and B, they're enriched for different chromatin compartment, and for example, U chromatins and the heterochromatins. And then TADs, they're conserved between different tissues and species. And then the disruption of TAD boundaries might have a severe effect in the phenotype and the gene regulation. And personally, I think that the importance for the test is really they are preventing the wrong wiring between the enhancers or even the repressors to their target gene. And then we also show you that you can use software to detect dynamic quantum looping events and to their target genes. And then one thing we really don't have time to talk about, there's again, single cell Hi-C technology is also emerging. And then if you're interested, I think when Shirley covered the single cell attacks in R6, you can do more research on the single cell Hi-C as well.